Good morning, everybody. I hope you're fresh after this wonderful evening yesterday. And before I introduce the speaker, I just wanted to thank Iqbal for the fabulous evening we had yesterday. I mean, what a treat. So, big thank you. And maybe thank you. So, today we have a fabulous program as well, and we're going to start with Professor Ute Maria Bandig from the Karolinska Institute in Stockholm. Professor Bandig is a PhD at the University of Hanover, which is actually quite close to where I grew up. So I've been in Hanover quite a few times. It's a wonderful place to start studying, very famous university for biochemistry. She then, after PhD, moved to a postdoc at the Karolinska Institute in Stockholm. Came back to Germany for an independent group leader position at the Helmholtz Institute for Infectious Biology, and then went back to Karolinska for faculty position and rose up through the ranks to professor position right now. Among faculty and prizes, most importantly, are probably the Hans Krebs Prize for a PhD, but she's also a member of the European Academy of Microbiology and an associate member of the ISNPC-15. And I'm really looking forward to the talk today, talking about Pseudomonas aeruginosa. And I think the title is too long for me to read, but I do leave the title to you.
Thank you very much for this nice introduction, and I also would like to thank the organizers and especially Professor Chowdhury and Dr. Yahan for inviting me, and also Comslet for inviting me. It's a pleasure to be here. We have long-standing collaborations in the subject that I will present today. However, I will present more the basic science work that we do, so the background work that relates to biofilm formation in microorganisms. So my talk today will be the physiological consequences of horizontal gene transfer of a novel cyclic dinucleotide second messenger signaling system into Escherichia coli. And so what is second messenger signaling? Second messenger signaling was actually discovered by Earl Wilbur Sutherland in 1967, and he got the Nobel Prize for this at some point. So he observed that the hormone epinephrine stimulates the liver to convert glycogen to glucose, and this conversion required a soluble intracellular molecule, namely cyclic AMP. So second messenger signals are produced in response to a first signal. They are usually small molecules that are rapidly synthesized or degraded by enzymes. And they are can also be released by ion channels. And there is a spatial and temporal regulation of this signal within the cells in order to promote signal specificity. Second messengers can be, for example, hydrophobic membrane-standing molecules like phosphatidylcholine inositol, but it can be also water-soluble molecules like cyclic AMP, cyclic GMP, cyclic AMP GMP, cyclic di-GMP, and all other cyclic di or oligonucleotides. But it can also be gases like CO or nitric oxide. So the broadness of second messengers is vast. So usually when we come now to bacteria, what are the components of second messenger signals? And these relates not only to bacteria, it also relates to eukaryotes. The first, but in all of these organisms, the principles of second messenger signaling are basically very similar. So we have first messenger, which is an environmental signal, and that can be as diverse as light or amino acids, for example. The signal can also come from inside the cell, from the cytosol, and it can report, for example, the nutrient or the redox status. But it can also be, for example, temperature. After the reception of this first signal, the second messenger is produced. And I will give these principles of second messenger signaling cascades with cyclic di-GMP, the most ubiquitous second messenger in bacteria. So upon the reception of the first signal, cyclic di-GMP is produced by enzymes that have a so-called GGDF domain, cyclic digonal cyclases. After production of the signal is sensed by a receptor. The receptor can itself then promote a response, a target response, or the receptor can promote a target response through protein-protein interactions. To shut off the signal, we need a degradation, hydrolysis of the signal, and that is performed in the case of cyclic di-GMP by so-called ERL domain proteins. A second messenger signaling is very flexible, as there can be a physiological response to a first messenger in the range of milliseconds, or it can be a response in the case also in the during hours. So we have a broad range of also of a time-dependent response on a second within the second messenger signaling system, and that is why maybe also that bacteria and also higher organisms use such a messenger signaling very widely. The common view on a prokaryotic signaling system was imprinted for a long time by cyclic AMP signaling in Escherichia coli. And cyclic AMP signaling in Escherichia coli is quite linear. Upon the absence of first messenger, which is in this case glucose, the adenylyl cyclase is phosphorylated by the phosphotransfer system in the inner membrane. This phosphorylation activates adenylyl cyclase to produce the second messenger, cyclic AMP. Cyclic AMP is then sensed by the cyclic AMP receptor protein, which is a transcriptional regulator. And this transcriptional regulator regulates on the Escherichia coli chromosome and the transcription of over 100 or more genes. So in this case, also the system is linear, and we have only one adenylyl cyclase. The physiological response of Escherichia coli is widely transformed by the second messenger signaling system. Usually, when researchers research on the signaling system, the cyclic AMP phosphodiesterase, the degradation of the system, is usually neglected, and also that cyclic AMP, for example, can be secreted in some cases, is usually neglected. So but some time ago, we discovered a novel second messenger signaling in bacteria, and that is a second messenger cyclic di-GMP. This second messenger system was discovered rediscovered only in 2004. And why was that? bacteria basically were considered to be too small to require complex second messenger signaling pathways. And also Stuart Schreiber already stated a long time ago, life would not exist with macromolecules alone. In bacteria, the focus was more on secreted small molecules and such as quorum sensing molecules. Cyclic GMP is a second messenger that occurs exclusively in bacteria and also in some eukaryotes by horizontal transfer by bacterial species. We have rediscovered cyclic GMP in 2004, and we discovered that it can regulate ubiquitously the transition between biofilm formation versus motility in Salmonella typhimurium and other bacteria. So upon high cyclic GMP levels, facility the production of biofilms and adhesive extracellular matrix components is promoted. And upon low cyclic GMP levels, which are promoted by the degradation by the ERL domain proteins, the phosphodiesterases, motility is promoted, such as flagella-mediated swimming, swarming motility, switching motility into the Monas aeruginosa, and also motility in a variety of other bacteria. So this is quite a ubiquitous signaling system, and its impact goes through gram-positive, gram-negative bacteria, and basically through all, mainly to the majority of bacteria throughout the phylogenetic tree. So what are biofilms? This is a developmental cycle of a biofilm, and biofilm basically are the multicellular aggregates of bacterial cells in contrast to a single cell, a single planktonic cell of the bacteria. So these biofilms are the natural mode of growth of bacteria and are also a huge clinical problem. For example, those biofilms are refractory to treatment by antibiotics. They have a huge high antibiotic tolerance, and they are also refractory to the actions of the immune system. So in some cases, biofilms, for example, can exist for decades in patients, or even almost for 50 years, as have been reported on biofilm infections in human beings. Those biofilms are more or less latent, not acute virulent, but they of course contribute to morbidity and mortality of the individuals. So besides the regulation of motility versus sessility of cyclic GMP, cyclic GMP also regulates the transition between sessility and acute virulence. Acute virulence, for example, is promoted by the secretion of specific virulence factors and by induction of the pro-inflammatory cytokines. Sessility, in this case, is related to biofilm formation, which leads to chronic infections in patients. So, what is special with the cyclic GMP signaling system? The cyclic a hallmark of the cyclic GMP signaling system is actually its abundance of cyclic diet GMP turnover proteins encoded by one bacterial genome. And that is called the cyclic diet GMP IQ. Within one phylum, there is usually a linear relationship between genome size and the number of cyclic diet GMP turnover proteins. And this slope is quite steep. For example, for gamma proteobacteria. Gamma proteobacteria can possess as many as over 100 cyclic diet GMP turnover proteins. Upon reduction of the genome size due to host adaptation, the cyclic diet GMP signaling system usually disappears. But in some bacteria, there can be still maintained one or two components of this signaling system. Cyclic diet GMP signaling in Escherichia coli, the topic of this talk. In this organism, the cyclic diet GMP signaling system is actually relatively modest. There are only about 30 cyclic diet GMP turnover proteins. So upon some observation that we did by genetic studies, we were then asking the question, what happens when, for example, into Escherichia coli, a novel cyclic second messenger signaling system comes in? And what this question was based on the observation that on the right border of a right border high pathogenicity island in the commensal strain Escherichia coli EcoR31, a novel signaling module was observed. And this signaling module actually contained a protein which is homologous to the dinucleotide cyclase DNCV of Vibrio cholerae L-TOR. So we were asking the question, does this novel second signaling module interfere with cyclic diet GMP signaling in Escherichia coli? So we first tested in this strain whether the cyclic diet GMP signaling system is still in place and working as it has been predicted for other bacteria. In order to do that, we overexpressed diagonal cyclase and we overexpressed a phosphodiesterase to see whether our biofilm model system that is here displayed by X, Y, and graph colony morphology is still regulated by cyclic diet GMP. Cyclic, and we could see that this is indeed the case when we overexpress the diagonal cyclase, we see an upregulation of biofilm formation. And when we overexpress a phosphodiesterase, we see a downregulation of biofilm formation. We then also looked at the motility and how motility is still regulated by cyclic diet GMP in the strain EcoR31. And indeed, we see also motility is regulated by cyclic diet GMP as conventionally reported. Upon overexpression of the diagonal cyclase, again, motility is downregulated, and upon overexpression of a phosphodiesterase, motility is stimulated. So we were wondering then, what does the introduction of the novel second messenger signaling system that is exemplified by the dinucleotide cyclase DNCV regulate biofilm formation and motility. And we could observe that biofilm formation and motility is regulated by the diagonal cyclase DNCV. However, biofilm formation and motility are downregulated by this novel second messenger signaling system. And this is shown on this slide here. When we overexpress the dinucleotide cyclase DNCV, we see downregulation of biofilm formation downregulation of cell aggregation, and downregulation of the expression of amyloid fimbria, one component of the extracellular matrix. On the other hand, motility is also downregulated upon overexpression of the dinucleotide cyclase DNCV, while when we delete the catalytic activity by amino acid substitution, we regain motility equally as biofilm formation. So in summary, we conducted some more experiments, like we also overexpressed and purified DNCV and characterized its catalytic activity. And we could see that DNCV is actually producing the second messenger site 3' cyclic GMP AMP and some other second messengers to a minor extent. And we think that DNCV producing cyclic GMP AMP is actually inhibiting biofilm formation through CSGD expression on the transcriptional level, and it is also inhibiting motility by interfering with the flagellar regulon cascade. So how does cyclic GMP AMP then exert its function? We always need a receptor. The receptor has been identified in Vibrio cholerae as the patatin-like phospholipase CAP-V, which is encoded upstream of DNCV. A proposed function of this patatin-like phospholipase receptor is the hydrolysis of phospholipid into free fatty acids and lysophospholipid upon binding of cyclic GMP AMP. Whether this is occurring also in E. coli, as it has been described in Vibrio cholerae, needs still to be shown. But when we initially characterized such a novel second messenger signaling system, we will usually ask the following questions. What are the mechanisms of cyclic AMP GMP to downregulate motility and biofilm formation in our case? What is the identity of the receptors? Is it the patatin-like phospholipase CAP-V, or are there additional receptors which play a role? Which enzymes degrade cyclic AMP GMP? And also, are there alternative targets besides the biofilm regulator CSGD and motility? These questions led us to a novel project, and that is kind of a serendipity in science or in a scientific project that I would like to describe for you. So not always when you look for a specific target, you will find maybe the solution immediately, but on the way, one can also maybe make a new project out of some side observations, or even of a new project made out of some one can call it mistakes or erroneous observations. And this I would like to describe because this is an excellent example of how a small error can lead to a novel project and actually to a novel perspective in science, and that is based on also the student being very observant in what he or she was doing. So we were asking the question, what are the mechanisms of cyclic AMP GMP to downregulate motility and biofilm formation? And in order to address this question, we were actually overexpressing the receptor CAP-V, but on the way, actually, we changed our research question to and found out that the cyclic AMP GMP receptor CAP-V Q329R, a variant of our original receptor, adapts a new functionality. And how we discovered this? So we overexpressed the patatin-like phospholipase CAP-V in a commensal E. coli strain in the model organism Escherichia coli K12, a standard laboratory strain, and we saw no effect on motility. However, when we overexpressed a variant of CAP-V Q329R, which was obtained by us by a cloning error, we saw a downregulation of motility. And we saw not only downregulation of motility, we also saw extensive filamentation upon overexpression of this variant. While the wild type did not cause extensive filamentation, maybe only a very slight elongation of the cells. So cell filamentation usually occurs upon the subjection of stress, and it is affecting the cell division. So cell division in bacteria is a process that can be divided into several steps, and usually we first have a chromosomal replication, then we have elongation of the bacterial cell and nucleoid segregation. Upon nucleoid segregation, Z-ring is formed in the middle where the cell will eventually divide, and upon invagination, eventually we receive then cell division in two new cells. So we could, when we looked in the mechanisms how CAP-V, the variant, is affecting cell division, we found that the FTS Z-ring formation is still occurring. However, imagination is inhibited and nucleoid segregation is only partially performed. So we have an inhibition of cell division by the CAP-V variant, beyond the formation of the FTS cell ring, which are the molecular mechanisms of this inhibition is still to be elucidated. Such a phenotype like extensive cell filamentation in a model strain of Escherichia coli can of course be maybe only an example of serendipity, and we observe such a phenotype only in one strain, and otherwise it does not have any impact. So in order to show or to investigate whether the effect of the CAP-V variant is also present in other bacterial isolates, we selected a number of bacterial isolates like commensal Escherichia coli strains, some virulent or pathogens, and also the pathogen Salmonella typhimurium, a close relative to Escherichia coli. We selected those strains as, when we looked in the databases, we could see that CAP-V and a cyclic GMP-AMP signaling system is mainly present in commensal bacteria. So when we overexpressed the variant in this panel of strains, to our surprise, we saw that in most of the strains, we still see extensive cell filamentation upon overexpression of the Q329R variant. And not only cell filamentation is affected, also biofilm formation is severely affected upon overexpression of this CAP-V variant. that nucleotide cyclase DNCV and this second messenger signaling system has been reported to affect susceptibility to phage infections. Therefore, we investigated whether our CAP-B variant is also affecting susceptibility to phages. And in contrast to what is reported in the literature for the wild type signaling system, we found that our variant actually enhances the susceptibility to, for example, the phage P1, that is a common phage which affects Escherichia coli. So upon only the substitution of one amino acid in a signaling module, we can actually revert the physiological response of the bacterial cells. And that is in a way surprising, but also shows us how flexible and plastic maybe protein functionality is. Patatin-like phospholipases actually occur in most bacterial species throughout the phylogenetic tree and also up to eukaryotes. But in bacteria, patatin-like phospholipases have been mainly investigated as virulence factors. And usually they are inactive in the bacterial cells and they are activated by host factors upon transfection into eukaryotic cells. One prominent example is XOU, which can be activated by monoubiquitination or polyubiquitination. So what is the physiological role of patatin-like phospholipases? A phospholipase is an enzyme that hydrolyzes phospholipid, the major constituents of the lipid bilayer membrane. And patatin-like phosphodieesterases are basically a subclass of phospholipases A2, and they hydrolyze the second ester bond here. The physiological importance of patatin-like phospholipases is wide. In eukaryotes, patatin-like phospholipases are associated with human disease like non-alcoholic fatty liver disease. So, for example, a variant isoleucine 129 methionine of the patatin-like phospholipase A3 is actually involved in such an inherited disease. In bacteria, on the other hand, patatin-like phospholipases can be virulence factors, but physiological roles of patatin-like phospholipases have been poorly investigated. I wanted to discuss shortly also biofilm formation in Shevanella algae, and I think I will have some minutes. We also recently extended our biofilm research and investigated biofilm formation in Shevanella algae. And why did we do that? Shevanella species are widespread, aquatic, predominantly marine bacteria. And importantly, Shevanella algae has been emerging as an occasional pathogen. Shevanella are known for their broad respiratory capacity, and as aquatic bacteria, Shevanella have a high complex cyclic GMP signaling system. And cyclic GMP signaling is also involved in sessility, motility switch, lifestyle transition in Shevanella. So our question was to connect the specific features of Shevanella and asking the question, does respiration of different anaerobic electron acceptors play a role in the regulation of biofilm formation? And indeed it does when we added, for example, the anaerobic electron acceptor DMSO or nitrate to our cultures, we see a strain-specific upregulation of biofilm formation in Shevonella algae. We then were asking the question, does respiration affect the regulation of biofilm formation? And indeed, to answer that question, we first need to know whether the respiration indeed occurs in all the strains that we have investigated. And indeed it does when we do assay to look to assess nitrate respiration in Shevonella. We see that all strains basically perform nitrate respiration. So the absence or the presence of these respiratory components is not responsible for the differential response we see upon the addition of anaerobic electron acceptors. In congruence with that, when we then delete the respiratory modules, we hypothesized that we abolish the specific biofilm response that occurs in the presence of the anaerobic electron acceptors, DMSO and nitrate. And indeed, that is the case. So here we have the DMSO reductase operon. And in this case, when we now delete this operon, we see, first of all, that the product is not made in this case. But when we then look at the biofilm response, we also see that the biofilm response in the case of without anaerobic electron acceptor, it stays the same in the mutant and the wild type, while upon addition of the anaerobic electron acceptor DMSO and the deletion of the respiratory operon, we see a downregulation of biofilm formation, and the respiratory response is basically gone. And the same happens when we look at nitrate respiration. This the upregulation of biofilm formation is accompanied by a very specific transcriptional response, and this transcriptional response is actually linear to also the amount of upregulation, to the degree of upregulation of biofilm formation that is occurring. So upon the addition of DMSO and upregulation of biofilm formation by this electron acceptor, we see the differential regulation of several genes. However, upon addition of nitrate, which causes a much higher upregulation of biofilm formation in the same strain, we see also that many more genes are differentially regulated. When we look at the overlap in this on the differentially regulated genes, we see that in the first run that maybe most of the genes are common to those two types of biofilm. However, when we then look at which genes are up and down regulated, we see that the overlap is not as large anymore, indicating that we have a highly specific response and a transformation of the physiology and the metabolism of the organism in response to distinct anaerobic electron acceptors. And here we just show, we can just classify the genes that are differentially regulated, and we find that metabolic and cellular processes are mainly up and down regulated. Our major question then was, of course, is there a role in cyclic GMP signaling in the biofilm response in the presence of anaerobic electron acceptors? And here we list in this heat map the up and down regulated cyclic GMP turnover proteins. And as you can see, there is quite a number of them, many more than in Escherichia coli. And we find that upon addition of DMSO, most upregulated is one GGDF gene, one diagonal cyclase, but upon addition of nitrate, there are two different diagonal cyclases mostly upregulated. There is also one diagonal cyclase most downregulated, and that perhaps indicates the differential biofilm formation that occurs in the presence of nitrate. We then verified that biofilms produced by Shevanella algae are indeed susceptible to cyclic GMP signaling, and when we overexpress a phosphodiesterase, which should downregulate biofilm formation, we see indeed downregulation when we have no supplementation, equally as when we supplement with the electron acceptors, DMSO, and nitrate. So cyclic GMP is most likely in many different ways involved in the upregulation of biofilm formation in the presence of anaerobic electron acceptors. We were then asking, because that is a physiologically and also ecologically relevant question, does surface incorporation of anaerobic electron acceptor promote biofilm formation? And indeed it does. So when we take a strain which is responsive to DMSO and nitrate, we see when we add the electron acceptor just into the surface, we see upregulation of biofilm formation. However, when we take a strain which is not responsive to those electron acceptors with respect to upregulation of biofilm formation, we see that the biofilm formation on the surface remains basically the same. This Shevanella genus is known to be present in the marine environment in several different water depths. And in the several different water depths, we find usually strains or species that respond differentially to the presence of electron acceptors that are present in the specific environment. However, with our research, we could now add another dimension to this, and we can state that we have a strain-specific biofilm formation that certainly then enhances the ecological opportunities. Since upon the presence, for example, of DMSO, which is produced by, for example, phytoplankton, or the presence of a 3-methylamine N oxide, which is, for example, produced by fish, Shevanella strains have two possibilities. When they are responsive to those electron acceptors, they can more readily colonize the surfaces of the specific higher organisms. While when they are non-responsive to these electron acceptors, they remain maybe as planktonic cells just in the marine water. In this case, we can divide, so to say, the ecological niches into two separate ecological niches and widen the ecological opportunities of the species. Yeah. So besides nitrate and DMSO, which are abundant actually in the marine environment, and they are substances that are included in the global sulfur and nitrogen cycle. We have several different other organic and inorganic electron acceptors that are accepted by Shevanella algae and other Shevanella species. So in this marine environment, there are a lot of opportunities for Shevanella to differentially form biofilms and occupy different ecological niches. And this broad variety of these molecular responses by Shevonella algae still needs to be investigated. What there may be also practical implications of this research. We can, for example, engineer bacterial isolates in much higher specificity, and we can engineer isolates to specifically colonize specific surfaces in order to perform maybe also in order to perform specific tasks such as to degrade pollutants. Last but not least, I would like to thank the once again the organizers for inviting me, and I would like to thank and acknowledge all the people involved in this work. The Shevonella algae work was done by Jonathan Rodriguez, and the second messenger signaling system characterization in Escherichia coli was done by Kenya Lee, and also Sulman Tafik helped with that. And then we had a lot of different collaborators. Without them, this work would also not have been possible. Thank you very much for your attention.
There's time for one quick question. One quick question over there.
Well, thank you very much for the nice presentation. So, the cyclic GMP and cyclic GMP, what is the unique difference? One or say pathway or different pathway?
There are different pathways, and there are distinct enzymes which produce cyclic di-GMP and cyclic GMP. Cyclic di-GMP is usually produced only in bacteria, and and eukaryotes produce another cyclic dinucleotide second messenger, which is 2' 3' cyclic GMP AMP. And this second messenger is actually at the heart of innate immunity in higher organisms, in metazoa. All those or many of those enzymes are actually are usually present derived from bacteria and evolved into higher organisms. So there is a phylogenetic relationship between some of these enzymes. And DNCV, the dinucleotide cyclase, is phylogenetically related to the enzyme in higher organisms that is producing 2' 3' cyclic AMP GMP for as an innate immune response component.
Let's thank, so one more time for fascinating job. Thanks for time for my questions to the public.
And of course, also get a present.
All right, I can start.
Yeah, well, I should give you an introduction, Mustafa.
Okay.
So, next week, Professor Mustafa Güzel from the Istanbul Medical University, Department of Medical Pharmacology, the professor there. Mustafa Güzel did his PhD in the United States and spent 14 years in the industry before he returned to Turkey for his academic career. I must admit, this list of acknowledgements is way too long because we're already above time, but I will mention that he's deeply involved in the COVID-19 response of Turkey and a number of advisory roles, and I'm looking to this talk.
Thank you very much.
As soon as it shows up.
Okay. See, it's truly a great honor to be here. I would like to thank the organizers and also ICBS team, the crew. This is my first time visit here, inshallah. It's not gonna be my last. So, Assalamu alaikum wa rahmatullahi wa barakatuh. Merhaba from Turkey. Good morning. Thank you for inviting me again. So I will stick with the plan, give you the flavor of this actually presentation. I aim to usually to satisfy mostly the youngsters, the graduate students. So there are a lot of pictures. I don't want to go into deep science, but there will be some science to maybe in sense satisfactory. So this is my life in the States. Usually after my graduate and I was a high school teacher 5 years, then after that I moved to States. These are my life cycle in the States: research assistant, research scientist, RQL, lab manager, research investigator at Transtech Pharma, High Point, North Carolina. Then moved to Turkey in 2014, assistant professor, associate professor, and full professor in this year. And then director of the Drug Discovery Research Center. Then these are the clinical candidates that I was part of the synthesis design. So I contributed in bringing these molecules to clinic. In my career, probably I synthesized with my hands, own hands, 12,000 compounds, but only 12 of them became a drug candidate. Not drug yet, not drug yet, drug candidates. So it's a lengthy, very tedious work, as everybody knows. Not easy task. So you will see some highlights of. Mostly, I will emphasize the cancer metabolism on my presentation, but most of these you won't see. Unfortunately, this right now I heard it was dropped in the phase 3 Alzheimer drug. This is the receptor advanced glycogen product. Yesterday, you heard some presentation. So, and I work on the diabetes area, GLP-1 agonist. This is in the phase 3 right now. This is kind of my baby because I was the one who synthesized the lead molecule and the drug candidate as well. So, whenever I see this molecule in front of my desk with my initials on it, and people are diseased subjects are using it, and it gives me this killing passion to make your own molecule and bringing it to the market, and still keeps me sleepless at night. So inshallah, and I have the similar outline here. I will give you the situation analysis, introduction of drug discovery, some cancer metabolism project, industrial production of favipiravir. Last year, I don't know if you heard some of my presentation. We'll give you some highlights of the synthesis of these domestic, and then natural medicine for COVID-19, some other projects, acknowledgements, and conclusion remarks. I have this bad habit, picture with the Nobel Prize winners. I don't know why. So I always insist to put this present on my presentations to inspire, especially the young students. Graduate students. This is E.J. Corey from Harvard professor. 90. This is my advisor's advisor. My advisor was a Harvard postdoc when he got the Nobel Prize. When E.J. Corey got the Nobel Prize grant. So, Sharpless. When this picture was taken. He didn't get his Nobel Prize, but he was kind of my idol because during my PhD years, I followed his footsteps. I emailed him humbly. He sent me back, "Thank you, Mustafa. I wish your career success." These are down-to-earth guys. So Nih President professor in 2010. This is the Martin Chalfie, Columbia professor. He is the one actually discovered the GFP, green fluorescent protein. Right now, it's pioneered a lot of research. This is the Brian Kobilke, GBCR, Stanford professor. He came to Turkey. And I don't know why I put these two pictures. This is the first time in lab, and I entered in 1982. We were trying to extract from the orange peels the essence of the orange in 1982. Now you figure out my age too. So this is the diabetes rat model, and when I started my diabetes project in inshallah one day I want to have a picture with inshallah one of your audiences, especially Professor Iqbal. That's why I left this empty here. Inshallah, one day we will have a picture with him. So drug discovery is a lengthy process. I don't want to go into deep, but I consider myself usually in this dark blue line. So as a medicinal chemist or a scientist, if you are bringing an IND, investigation new drug, for us, that project is finished. The only thing you have to do the backup compound as the same safety, same efficacy. Then your job is finished. So you have to find or investigate the destiny of the safety and efficacy, and then if you are lucky, you come to this one dot. If you ask me, Professor, do you have any one dot here? I don't. I mean, I don't consider favipiravir my molecule. It was a generic. So, but we have some candidates around here. 12. You saw this in the slides. So I like this slide also. Maybe living in the east side of the world should also we should strategize ourselves. These are the global companies, R&D expenditure. These are $1 billion, by the way. So $58 billion. AstraZeneca spent 15 years on R&D. Only how many molecules brought to market? 5. So that's which one? It's AstraZeneca, right? Sorry, the Amgen. Amgen spent half of what they spent, $33 billion. And how many molecules they brought? 9 molecules. So this tells you that how strategically we should move, or we should realize in terms of drug discovery and what should we do. Of course, another important thing, how much it sells, the profit. It's also important. So that's a slide. I like it, especially in Turkey, in Pakistan, what kind of indications that we should move. Like, for example, Leishmaniasis or dengue virus is prevalent here, right? It's very common. So then you should prioritize your drug discovery and development efforts based on the indication. So we should realize also 90% of the clinical candidates unfortunately fail to reach the market. That's a fact. So 9 out of 1 compound cannot make it to market if you bring it to the clinic. So and average estimate cost of the bringing 1 molecule to market, we know that it's $2.6 billion. However, if you look at that slide, some numbers sometimes it doesn't translate. When you divide this, it's almost $10 billion. That's why medicines are so expensive, especially when you see in the COVID-19, the Paxlovid, I believe it's a $700 1 box. It's the COVID-19 medicine. So as a medicinal chemist, I consider myself one. We should realize most compounds do not become medicines. And I gave you example, 12,000 compounds with my hands I made, but only 12 of them became a drug candidate, not drug yet. So because the complex disease targets, body adverse reaction for absorption, and it cannot be scalable. Sometimes you make a molecule like Taxol, you know, and you have to probably bring 40 steps to bring the molecule, the Taxol. That's why the Taxol is still an expensive drug. So as an academician, I tasted both academia and the industry. So we can do a lot of rare cancers, infectious diseases, enzyme activity, assay development, heat discovery. We can train and publication in vivo efficacy if you are lucky, training. But this is the bottom, the yellow line I want to emphasize. We have to partner with pharma. If we cannot partner it, and I gave you example the other day, Lyrica. Richard Silverman tried to bring that to market for 15 years. He was greedy, he was Jewish American greedy professor. He said, I wanted to bring this market by himself. 15 years he struggled. He couldn't. He licensed to Pfizer. Now it's became a drug, blockbuster drug. Last year it sells at $5 billion, Lyrica, pregabalin. So it's not easy. Even if you're a professor in Harvard, MIT, it's the same thing. You have to partner with pharma. Without it, you cannot manufacture, market, and medicine. Even in the iccbs, it's the same thing. So you have to commercialize or market it in a sense with a pharma company. So I'm moving to here to drug discovery. With this, without discipline, this you cannot have the medicine also. So you have to bring all these experts. In Turkey, we struggle the same thing. Everybody's becoming a professor. Oh, I became a professor. Now I know everything. No, you don't know. You just started a football match. You are still 0-0 if you are trying to develop a molecule. So biochemists, cell biologists, bioinformatics, as you can see, in vitro, in vivo pharmacologists, and clinical pharmacologists. If you have a clinical data, 5,000 patients, if you have a strong, if you don't have a biostatistician, you're screwed. You just look at the data, and data will look at you. You cannot even understand it. So my passion in the cancer area, it was when I started this project, was the cancer metabolism. What is cancer metabolism? I want to emphasize this. In 1930s, Jewish German Otto Warburg postulated or hypothesized with or without the oxygen supply, cancer cells ferments or survives in a different way. And he won the Nobel Prize, and he was the one actually nominated Nobel Prize in 45 times. And he was believed also he won the Nobel Prize in 1945 because of the Hitler Nobel committee denies it because of the World War II. So he won the Nobel Prize in 1931 because of his findings. What is he telling? He tells basically normal cell, as you know, the glucose is taken, glucose phosphate, and then pyruvate. After pyruvate, it goes to the Krebs cycle, as you know. But in cancer cells, he postulates it doesn't go to the Krebs cycle. I mean, cancer cells are not that dumb. I mean, this ATP is our energy, right? But they actually proliferate in a different way, much faster. So this is 2 ATP. This is, I don't know, I forgot my biochemist. I think 36 or 38 ATP. I'm sure there are some biochemists. So, but this is much faster than normal cell proliferation. So this is what actually gained the Otto Warburg, that experiment. When you look at this normal tissue, animal neoplasms and human neoplasm, you can see an anaerobic and the respiration. So basically, this is called Warburg effects. So our project was, can we reprogram this? Can we change this? This is the Wikipedia from glucose to pyruvate. That's the fate of the, when you take a sugar from, you know, anything that carbohydrate we ate. Last night we ate a lot of carbohydrate rice. It was very nice, by the way. And this is the glycolysis pathway. These 3 enzymes that I want to highlight, I cannot, yeah, these 3 enzymes that are the reversible and the delta G. When you look at it, these are 3 enzymes that highly work, that the red ones, pay attention to those. I work in that area in the United States. I work in the Turkey in this one, never work in the PFK. These are irreversible steps. They have the delta. When you look at the delta G, you will understand. So this is what we work in this one. Of course, there are some other targets in the cancer metabolism. This is the science article, Science Advances. PI3 kinase, fatty acid synthase. Yesterday we had lipid metabolism, some other glycolysis pathways that you can target. This is a hot target in the cancer area. So hexokinases, there are 4 of them. Hexokinase 2 is overexpressed in the cancer cells. Sometimes it's believed to be 10 to 15 fold. That was our hypothesis. Can we, without touching the living cells, living tissues, without harming them, can we stop tumor growth? And that was the process. So this is the hexokinase biochemical transformation. I don't want to bore you with the biochemistry too much. This is the assays that we developed: tumor cell cytotoxicity, lactate production, ATP, mitochondrial and membrane potential assay. Of course, without the nice pictures, you have to find the allosteric binds or active binding site of the receptor. With the ligand, this is the ADP binding site of the hexokinase 2. So we developed a molecule based on that. There are 4 players on the market that bring to clinical or preclinical stages: 2-DG and the 3-bromopyruvate, lonidamine, as the inhibitors of the hexokinase. Methyl jasmonate. In the United States, we made a lot of derivatives of this lonidamine, and we had a clinical candidate. We licensed it, 3,000 molecules. I'm not going to go into the details. In Turkey, I made the derivatives of the methyl jasmonate, which I'm going to quickly highlight it. And we made a handful of molecules, around 24, and we made much better than the methyl jasmonate analogs and screened them in a different cell line. And when compared with the and patented that highlighted with the different cell line, Niapaka as well. And also we tried some in vivo study. This is the molecule that I made in the United States. This is what actually makes me sleepless at night. When I see this in vivo pharmacology results, then you believe in your project. Why? Because let me explain this slide to you. This is data, 30 milligram per kg IP dose intraperitoneal. What we do, we make the subcutaneous cancer of the mice, and then we inject our molecule. This is vehicle, no drug. This is daytime in the 30 milligram per kg dose. And of course, we compare this with the gemcitabine, 5-fluorouracil, Taxol, anything you can remember as a cancer blockbuster agent. So this animal, unfortunately, at the 15th day was extirpated, terminated, and this animal, even the 33rd day, I saw it with my eye, because the vivarium guy, he was a Ghana PhD from Russia. He was my friend. He took me disguisedly in the lab. I saw the animal with my eyes. So then I believe that it's working. So we did the dose escalating study with this, and you can see that we found the effective dose, ED50, and therapeutic dose, and also LD50. We determined the LD50, and then of course overall you have to have an oral bioavailable drug. Nobody wants injectable like in insulin, right? In the medicines, this is really important. It has to be selective. It has to be oral bioavailable. Enzyme selectivity is really important. This is the profile we prefer in cancer metabolism. This is the patent we have in the United States, and also we patented the molecules in the in Turkey once. We also worked in the propolis derivatives for GBM, and the GBM is an unmet medical need, as you know, and there is no medicine when you look at it in the timeline, and when you look at it only to bevacizumab, they say it works. Temozolomide is a killer; it doesn't work. We found this in my lab. I said, by the way, this project, I did not receive any money. I had the compound in my hand. Why don't we make analogs of it? So from the scratch, we made the project. No money from any. So we made this molecule, derivatized, and screened some of them, compared when you look at the escape and the number 10, this is the number 10 molecule, we increase the tenfold activity in aggressive cell line. This is LN229. We did also other cancer cell line. So it was, this is published and patented work. Hopefully, we will move forward. But we are at the beginning of this. For some reason, I cannot control it. It goes really fast. I apologize for this. So these are the projects that I try to do in Medipol University, in the cancer metabolism, metabolic disorders, the diabetes, obesity, autoimmune diseases. And these are the targets. What can we do? Drug discovery, identification, lead optimization, drug development, SAR, pharmacokinetic and pharmacodynamic properties, and ADME properties. And these are the projects that, some of them completed and some of them ongoing. I'm not going to go in details. And about some reason, some of the my slides are skipped. And this is the team, and I consider the group in split. We have synthetic guys, in vitro, in silico, in vivo, and some natural products. These are the scientists in my lab that currently affiliated with me. And we have some also collaborative projects in with the Koç University, and Aziz Sancar won the Nobel Prize in cryptochrome, circadian clock. We found that one molecule that actually helps in that mechanism. So right now in Nature Communications publication, we also patented that out. This is the publication that also with the United States researchers. And I want to emphasize quickly, how many times MK we have? How much time? I don't want to.
Just go on, I think
I'm sorry?
Just go on, I have plenty of time.
I have plenty of, okay.
I'll stop you, don't worry.
All right, okay. All right, thank you. So this is the synthesis of Previr that I want to quickly some quick pictures. So when the COVID-19 projects or the COVID-19 hit the world, became a pandemic, we started thinking in Turkey what medicines we can see help in. This was with the TÜBİTAK affiliation project. TÜBİTAK and Turkey responded quickly. And whatever the molecule was in hand, we said, okay, we have to quickly synthesize and scale up. So Favipiravir was one of them. Hydroxychloroquine, I know it's out of the picture right now, but at the beginning of the pandemic, even in the United States, it was hot molecule. Remdesivir, that's the first molecule that, as you know, is the first molecule FDA approved. So we synthesized all three here. We actually stayed in the hotel, didn't go our home because most of them were using public transportation. So we stayed in the Atabike, the company's hotel, and we 12 of with my researchers, most of them were postdoc researchers, and they were assistant professors. With the bad times, we stick together and we synthesize all these large scale. So this was a true industry and university collaboration. We developed a process, we optimized that method, and this is the synthesis of Favipiravir. I'm not going to go in detail, but in Turkey, some people didn't believe in it. Oh, you brought from China or India this molecule, and you're tricking us. So I asked my assistants, look, take every picture of intermediates. Otherwise, we cannot convince them. So I did. These are the pictures of all intermediates, melting points, at least, so that we can convince the decision makers. Look, we can make this molecule in-house, no problem. It's not a rocket science. So from the sixth step, this was actually tedious one, because in this large scale, you have to scale up this one in one pot. Of course, in industry, we hate columns. We don't want to do column. You have to do crystallization. Column means a lot of solvent, a lot of silica gel. So, so without column and salting out with the sodium, ammonium, and this salt, we can make this compound right now 3 ton in month with the industrial capacity. So that was the so from so I apologize. So with this, with the 100-gram scale, we can make around 40-gram favipiravir. So the yield is acceptable. So this is what we finished, and then we got the licensing. Now affiliation group gives this medicine, distributes to the COVID-19 PCR positive cases. And these are the proof that the comparison with the NMR, proton, carbon, even the fluoro-NMR, and then maybe I should go with these instead of going sticky. And one-part synthesis, overall yield was without purification, extraction, filtration, and filtration. We got 43% of the yield, and this is the licensing from the Minister of Health that we got our product that can be given to patients. And these are the results that 8-step synthesis route can be finished in within 40 days. We finished this, optimize the yields. So the monthly capacity, as I mentioned, 3 to 4 tons. So the industrial scale-up production and alternative synthesis methodology still, of course, science never ends. It's still ongoing. And these are there are some Turkish picture, the Turkish words, so don't worry about it. I will translate it. So my assistants and me took like a photo album. So I want to share some of the photo albums with these starting the projects, entering the lab in the heart of the COVID-19 when everybody was locked down. We actually were in the lab day and night, actually working with my assistants. Even myself, you can see this was the Father's Day. I don't want to agitate myself, but in the Father's Day, I'm away from my kids. What can you do, Mustafa? Go become a role model for your students or your postdocs. So I went in and I synthesized 3 grams of favipiravir myself. And you can see my picture on the right corner. So, and this is the team. And without them, of course, we cannot be successful. With the Atabay facility, so we were able to finish within a quick time, scale up this molecule. The president of Erdogan recognized this. He invited us in all the TÜBİTAK COVID platform. We have 8 vaccination professors and 9 drug discovery projects. So all the professors were invited. Then later on, he invited us to his palace. Privately, we discussed 1 hour, 15 minutes. So he recognized the effort that we did with the favipiravir, with the SLA emergency cases. So I appreciate his support in terms of drug discovery. And these are the social media links. And if you are interested, you can click on the links and look at it. And in the natural drug discovery, we had second projects in the COVID-19. So my inspiration is also not just synthetic chemistry, some natural product driven compounds. Of course, there are a lot of natural remedies in the COVID-19 treatment. Quercetin is one of them, curcumin, a lot of them. Even the garlic diallyl sulfide is known. And here are the examples: genistein, indole, methanol, and also black cumin, the thymoquinone. As you know, right now we are trying to do phase 2 studies with the American company in Turkey with the COVID-19 treatment. They patented out, they got the FDA approval for phase 2 trial. So we are trying that also in Turkey in Medipol. Curcumin is another one, and allicin, as I said, and thiamol, carvacrol, and quercetin is another one, enzyme modified isoquercetin, because the cell uptake increased 40 times when you compare it with the quercetin. So these are good natural molecules for COVID-19. Boswellia, Artemisia annua. So we picked Artemisia and Arctine Lapa. And in Artemisia, we isolated from 5 kilogram. We did 20 different isolation conditions. Then we found some good cell proliferation and neutralization assay and the COVID for SARS-CoV-2, and published in phytochemical analysis recently. I don't want to go into detail about this, but we also screened some of the 20, 30 natural products and some drug repurposing candidates. We found some good molecules. Right now, this publication is unpublished results. I am right now writing this manuscript because in Chinese medicine, and this Artin, Lapa, and Artemisia was used heavily with this regimen. So we thought maybe we can also use this Artemisia and Artin, and as you heard from the Artemisia presentation the other day, Artin is also known. So we made also some analog, as you can see here, and in the right side. We did some essay, and right now we are trying to publish this soon, hopefully, and compare some results with the arginine and Artin. And these are good natural products that can show some SARS-CoV-2 or antiviral properties. And another projects that with the COVID-19 was the drug repurposing project. Montelukast, as you know, this single layer is the asthma medicine, asthma. So we screened from 9,000 FDA approved and in Turkey approved, EMA approved medicines, and screen them and look at the SARS-CoV-2 main protease inhibition. And we found the Montelukast was really proliferate 24 hours to SARS-CoV-2. And we published that in the molecular therapy. This is the good journal. I think impact factor is good, or 11 or 12 right now. And we registered in the clinical trials. In phase 2 studies, unofficial results, we haven't published the clinical results yet. 250 patients are tried. We compare with the favipiravir. We did a double-blind placeholder study. Among these 250 patients, none of the patients died. Unfortunately, favipiravir side, one patient died. So the results from the Montelukast on the critical was really good. So we are hoping that we can publish that clinical results in a good article, hopefully. So this is yesterday we talked about the Leishmaniasis, so I wanted to bring that also. I worked in the Leishmaniasis, especially in the BINAP analogs. This was the COST European grant project. I also published with my colleague in Molecules and Frontiers in Nanotechnology. I was the one synthesizing the analogs, and he made the emulsions and the phospholipid nanotechnology molecules. So our aim was to increase the, again, cell uptake, and amphotericin, as you know, is a problem, especially in the drug resistance. So there is an urgent need in Leishmaniasis medicines. There are some new liposomal amphotericin B analogs, but still it's a need new molecules. So we found one candidate we're trying to push forward. At the end of my speech, always we give thanks to my team. This is Istanbul Medipol University, my Güzel research group. I have 2 assistant professors in my group, 1 postdoc currently, 12, 14 PhD students, 2, 3 masters. We have formal groups. Actually, we have right now 8, 9 assistant professors who are trained in my lab, and still 5, 6 postdocs. They went to move to industry after they got their training. And we have a lot of undergrad students. Most of them actually they are medicine students, School of Medicine. They like to be in the drug discovery and development area, and most of them are gifted students. Among the 2 million entrants in the Turkey, they are the top 50, like first 50%, 50 ranked. So we thank them. This is just the first, just a small picture of my group. Some of them are missing. And Professor Iqbal visited this research center. Unfortunately, I wasn't there. This is our research center. We always thank them. We are open always to new youngster scientists who are eager, like they didn't lose the passion, like me. So we are always hiring with the projects. So please do send me your CV. We always appreciate the collaborators. Hoping with the Professor Iqbal and Hina, we already initiated some collaborations. Arshad from England, former TUSAP president. This is NIH president in Turkey. He is the professor in KTH and King's College. Georgetown professor Aykut, Bill from Wake Forest. So several collaborators, we thank them. We thank the finding sources, Medical Runner, Bob, Sci Life from Sweden, COST, the European Agency, and TÜBİTAK in Turkey. Without their support, of course, we cannot do it. This is Bosphorus. I told you I had a lot of pictures for the. So this is the Bosphorus. This is the Medical University, very close to Bosphorus. So anytime when you're in Istanbul, please do welcome to visit our school. This is the school's vicinity. This is the Istanbul Medical University's International School of Medicine and Center for Drug Discovery and Development. And I want to thank again organizers and inviting me. Last word of advice here: science is a three notion. Those who learn the first section became arrogant. Those who learn the second section will be humble. Those who learn the third section actually realize that they don't know anything. This is Qadi Ibn Tayyib's work. And the second one I want to emphasize, the science and technology are like two wings of the bird. Societies that can use the words, these two wings can easily fly and gain their freedom. Those who cannot fly become chickens. The chicken society does not even realize that their eggs are taken from behind while taking a handful of bait thrown in front of it. And I hope you get this message. This was the Charles Darwin message, but I use this heavily. And I sincerely, again, thank you for inviting me. I hope it was not much more boring. I didn't want to too much scientific and with the pictures. I wanted to give you the flavors of what we do. And thank you again listening to me. And thanks for the invitation. I hope I used my time.
Yes, thank you so much for all fireworks of ideas and molecules. I really enjoyed that. And I'm sure there are questions. And thankfully, we've got 3 minutes for questions. So
Okay.
please.
Thank you, Mustafa, for the whistle-stop tour of drug discovery and more particularly for the philosophy at the end. You spoke quite a lot about drug repurposing, which is a very valid thing to bring to our attention.
Mm-hmm.
But on a sort of different side of repurposing, recently target repurposing has become a theme for neglected tropical diseases, particularly Leishmaniasis.
Yes.
So I want, particularly as you spoke about glycolysis, maybe you could give us a view on drug target repurposing and the potential in neglected tropical diseases in particular.
Good point. For the glycolysis part, of course, I never worked in the Leishmaniasis aspect of it. Yes, especially when you look at the approved medicines, a lot of, for example, I can give you one example, filibanserin. This is SSRI, serotonin reuptake inhibitor. There was one Raleigh-based company when in America Viagra hit the world. Then a woman said, "Okay, we want also similar medicine." So there is a pink pill. They call it pink pill. When you Google it, you will see it. There's a company. They wanted to push the FDA. FDA rejected 3 times. I don't know if filibanserin is really. It says with the drug repurposing, if it's work or not. But FDA rejected 2 times. And then the 3rd time, FDA approved that because of this civil organization, the ladies' organization, I call this dark side of the pharma industry. Unfortunately, there are some. If there is a money involved, so FDA unfortunately had to approve the drug. The next day, the company sold $1.5 billion. So that lady, pink lady, went out. Probably she's in Marshall or Seychelles Islands, retired. So there are some direct repurposing, but if it works. So especially in the neglected, I mean, when you look at the database, 9,000 approved medications. When you cluster them, classify them, using now, we have magic words like artificial intelligence, data mining. So now global companies right now, believe me, they are into these rare diseases. SMA, for example. Right now, the Novartis is Zolgensma, making $2 billion. This is an enzyme replacement therapy, right? It's one shot is $2 million. And they don't budge, by the way. It's $2.2 million right now. They don't make it $2.1 million. Even in Turkey, there are some patients, they try to raise money because they are poor. And they are trying to get the money. They are short $100,000. They cannot get the medicine. So the global companies also saw this window, and they using the utilizing the artificial intelligence, clustering them, data mining, using the machine learning, even like in the social media. Oh, I took aspirin, now I had a scratch on my elbow. And they grab these keywords. Okay, aspirin makes or scratches this, and then they pick these ones and look at this. The global companies uses this artificial intelligence excessively. So using this, I think we can probably improve or increase the or look at the different window for the neglected diseases. This is, I mean, we should do, but maybe the governments, because still it's looked at as a not profitable way. Still some companies look at it, but governments also has to be participating. Maybe give you some incentives. If you make medicine, I give you like a grant. If you make medicines for neglected diseases, this is what I look at the approach. But it should be done in a smart way, not the dark side of the what I give you example.
So I think there's plenty of time to discuss this during the tea break that is going to start in a moment. And before we thank the both speakers, let me just tell you that we'll be pleased to be back here at 11 sharp. We're going to start the next session. Enjoy the tea break, and please join me in thanking both speakers for an excellent session.
Thank you.
Ladies and gentlemen, may I have your attention, please? I have an announcement. There is a tour being organized for the foreign participants to the new city, which is Green Sustainable City called Barrier Town, and this will be followed by dinner at the same in the same city. The vehicles will take you to the new city around 6:15 from the guest house. So kindly arrange your stuff accordingly. Thank you so much.